All right, so today's lecture is all about transport proteins. So these are going to be the proteins that play a role in helping your cells to be able to carry materials across them. And we're going to talk about each of them and kind of what they do. So the first type of transport protein is actually called a channel protein. So a channel protein is going to just help to move solutes through. So remember, a solute can be anything. It can be any type of like solid or molecule that's in your cell environment. As we're trying to move them through, we're actually going to move them through about 1,000,000 of them per second. So this type of protein in particular is also a transmembrane protein, which means it actually goes through the membrane. You see how this looks? These proteins can be open or gated. That's because, like anything else that we're doing, we need to be able to control when we can move items in and out of it. And the thing that's going to be the key to open these proteins are called ligands. So ligands are going to essentially just be the name of the molecule that's needed to either keep the gate closed or keep it open. In particular, this image is looking at channel protein. So just like a channel, think of a hallway. If you're having trouble kind of orienting what that would mean, think of a channel protein as being similar to a hallway. Okay, I'm going to try to write this out a little bit. So the same way a hallway allows you to go from a room that's here to a different room, down the hall, that's going to be exactly what a channel protein does. So another type of specific channel protein is called an aquaporin. So an aquaporin, like the name suggests, if you know your synonyms, you'll see here, as aqua tends to mean water, right? H2O, aqua. So an aquaporin, as you can imagine, is going to be a water channel. So it's going to be a channel that only works to move water through. That's because we talked about osmosis already. We talked about the importance of equaling or having the water being in equal quantities on both sides. So the aquaporin helps to make sure that we can move water in and out. It's going to be different than something like steroids or carbon dioxide or oxygen, which can go directly through the membrane. And if you remember, this is an example of simple diffusion, right? Because it goes, again, through the membrane directly. All right, so another type of carrier protein that we have are called transporters. So there's different types depending on how many solutes they carry in which direction, but these transporters are going to undergo something called a conformational change, which maybe you haven't heard of that term before. But a conformational change is going to have some type of, think of it as a shape change. So a conformational change has a shape change. So transporters are going to bond solutes and undergoes that shape change like I just mentioned, right? There's going to be some type of change in the shape. Where does this happen? This happens in the membrane, okay? So all of this will happen in the membrane. And why does it change? Because it needs to allow or be able to offer the solute to be able to go from one side to the next. So we want to keep it closed because we don't always want things going in and out of our membrane, all willy-nilly. We have to make sure that we have some control over it. So it's going to help to allow certain solutes through, and it kind of helps with the control aspect of the entire process. These transporters that I'm going to show you in a second, they're going to be named according to number and direction. So number and direction. And you're going to see that in a little bit. It'll be very, very clear. It's named according to the number of solutes they have plus the direction of those solutes. So kind of going back to our old days of structure determined functions, there are going to be two features that are found within these transporters. One of those features is going to be a term that you've kind of heard before. This is not the first time you've heard that term. And that term is going to be a hydrophilic, so love, philic pocket. So ask yourself, if this was a transporter, which it is, why does it need to have an area on the inside that loves water, right? You're trying to get this solute through to the other side. Why does it need a water-loving pocket? I hoped you told yourself, well, if this solute is on the outside, that means it is already hydrophilic. It already loves water. It already is polar. So if it goes through the membrane, which is hydrophobic or water hating, it's going to need some type of safe zone that it can jump into so that it can easily go from one side to the next. So that's why we have that hydrophilic pocket. And again, it's going to switch back and forth between these conformations of being open or being closed. So let's get into the first transporter. The first one is really simple. It's called a Uni Porter. So you kind of have an idea already of how many solutes this can carry. And it is going to be able to transport one solute, and they're actually going in the same direction. So one solute in the same direction. You see this is one purple solute, and this transporter is only going to allow things to, let's say, go inside the cell. It doesn't work both ways. It only goes inside. It only goes in one particular direction. So a Uni Porter, one solute, same direction. Symporter, I'm sure you can already see the difference in colors here, so you probably have an idea of what this one is going to do. But the symporter is going to be where we have two or more solutes, but again, they are going in the same direction. So in this case, you have the red ball and the blue triangle. There are two, right? There's our two. And they're going in the same direction. They're going, let's say, into the cell. So this transporter is going to help two or more things go into the cell. Finally, our antiporter is going to, again, still kind of keep that theme going of two or more solutes. But this time we're going in the opposite direction. So it can hold up to two or more solutes. In this case, it has this blue sphere here, and it has this magenta rectangle. The blue sphere is able to go, let's say, in, and the magenta sphere is able to go out. So this transporter, as you see, it kind of has two pockets. So there's a pocket there, there's a pocket there. That's because it's able to operate with two or more solutes. So to kind of just reiterate this active transport a little bit, I want to just kind of introduce you to one slightly new concept. I won't talk about it too much. It won't be on the exam very heavy, but I still want to introduce it. Again, remember our active transport. Nothing new has changed here. So it's still going to be able to use energy. That's standard. It typically goes against the gradient, meaning it's going the opposite direction, so it's going from low to high. And we've been talking about primary active transport. So it uses that energy we talked about via a pump. So it just uses some type of energy to kind of pump something from a low area to a high concentration. But secondary active transport uses an existing gradient. So if something is already moving from, let's say, high to low, right? So let's say there's a wave crashing, right? You open a door, all this water flows in. Something that is actually moving from low to high can hijack that system. And as that wave is crashing in, it can kind of just ride the wave and go along with the item that is going from high to low. So it almost like a piggyback of the existing gradient. Okay, so let's talk about channels for a little bit. Let's talk about these intracellular channels. So far we talked about transporters that allow an item to go from one side of the membrane to the next. But now let's talk about how we can get items between cells. So let me give you a little bit of a visual that might help better than my laser pointer. So far we've been talking about a cell membrane and how we have these different transporters to allow items to go in and out. Now between intercellular channels, we're talking about how cells communicate. So you have cell number 1 over here. How does that interact with cell number 2? Think about like a nerve cell, right? Getting something from one cell to the next. This is how that works. So I'm going to briefly just kind of introduce you to some of the terms here. A gap junction. So a gap junction is just a intracellular channel between animal cells. So this is important that you associate this with an animal cell. So again, gap junctions is animal cell. You do not find a gap junction in a plant cell. Plasmodesmata, I'm sure you can kind of get the gist here. This is going to be found in a plant. So this is going to be the channel found between plant cells. So plant cells is where that plasmodesmata is. All right, I'm going to introduce another kind of concept. Again, we're keeping it in this whole transport idea. Exocytosis and endocytosis. So this is the movement of large molecules in and out of the cell. So as you see here, exocytosis is basically what happens when you have something exiting the cell. So as we exit the cell, we call it exocytosis. The same way the term exit means leave, it's going to be the same exact concept with exocytosis. So that's when something is going to exit the cell. Let's say you're releasing some fluid. Let's say you're releasing molecules into the environment. All of that stuff is exocytosis. Endocytosis, EN, is going to be really similar to when something enters the cell. So this could be you eating something as a cell, your defense mechanisms you have, or even some things like invasion with bacteria, right? When you're getting a bacterial infection on your cell, this is going to happen through endocytosis. So there are 3 different types of endocytosis I wanted to briefly introduce. Real simple, just mostly definition based. In here, you can see the first one is phagocytosis. So phagocytosis is going to be cell eating. So when a cell eats something, it's going to be phagocytosis. So that's going to include more like a solid particle, phagocytosis. Penocytosis is where cell drinking. So think of, I know y'all might not be of age, but there's a drink called like Pinot, right? It's a wine, Pinot Grigio. And think of penocytosis, Pinot Grigio, that might help you remember it's a liquid. And then finally, cell coated. So if the cell is able to pick up something on its receptor, because it is coated in these receptors, we can call that receptor-mediated endocytosis. So RM, receptor-mediated endocytosis. Again, these are just kind of things, maybe multiple choice-based questions, just real kind of general, big oversight view things for you to consider. And then finally, my last slide is talking about cell junction. So how do our cells stay glued together? And there's basically 2 different types of ways. One of them is called an anchoring junction. So this is going to basically link the cell together, not only with each other, but also helps to link the cell to, let's say, the extracellular matrix or even some cartilage. So think about your different joints you have in your body. They're going to have a lot of anchoring junctions because you want to be able to link those cells with, let's say, some cartilage you have above that, right? That type of connection there is going to be an anchoring junction, like an anchor from a boat. The other type of junction that's very important is called a tight junction. This is probably a lot more common. This is going to help to prevent the leakage of materials. So the best way to think of it is like a seal. So it helps to seal certain parts of your body in those parts of your body, right? So for instance, you don't want items from your stomach, which has a lot of stomach acid, to then leak over into other parts of your body. That is not good because there's acid in your stomach and that's really bad. So you don't want that to happen. Your intestines have a lot of tight junctions because there's a lot of bacteria, a lot of cellular waste, body waste. You don't want that to leak into your body. That can cause a lot of problems. So that is going to be called a tight junction. So this wraps up the section on cell transport, cell channels, cell junctions, all those components. It's not too long. Please make sure you watch it, you review it. And this actually wraps up all the material associated with chapter 5.